Next week, though, you're going to have an exam on Monday and a final practical on either Thursday or Friday. And in the intervening days, that's as I say, Tuesday and Wednesday of next week, I'm going to offer the lab up for you to study. I'll be at room 304 during that time, so that if you want to come in and look at models or the SINDAVR, you may do so during those times. The other thing I want to talk about is so we're done with quizzes now. And first thing I want to say is that the way that I've divided this last group of material is that so down on the main campus, they started out with nervous tissue. They went through all the brain, all the spinal cord, and then the somatic and the autonomic nervous system, which is what we're about to do. And there's just one exam. Right? And so that exam is really hard. You can see some of that difficulty in this quiz today. The material on the brain is pretty difficult. So that's one thing that I'm doing to help you on purpose. The other thing is that because we have 8 quizzes, what the policy I have is that I'm going to drop the lowest quiz. The way the Brightspace gradebook is set up is that it treats the quizzes equal to the exams. So you don't look at that. It's a messed up. The grades are accurate, individual grades are accurate, but the way that it calculates it is not. And I think I figured out how to change that, but unfortunately I won't change it for you guys because I don't want to mess it up. So what I'll do is I download it into Excel or into Google Sheets, and then I do the calculations there. What I'll do is I'll separate the quizzes from the exams. The exams are the exams, and there'll be 5 exams that will be equally weighted. The quizzes, I'm going to drop the lowest quiz grade that you have, and I'm going to make your grade, your average quiz grade, the best that I can. Whatever it is, however I can do that, without actually just arbitrarily adding points to you. Hold on a second. After I'm done with that, making your grade as best I can, your quiz grade, then I will treat that as an exam grade, and so your final grade for this class will be out of 6 exams. Question?
Wait, so Brightspace increases our grade or like makes it decrease?
I don't know. I have never bothered to really see. It definitely affects it. It probably lowers your grade because all of the quiz grades, if I understand it correctly, it might be treating the quizzes equal to the exams, but I'm not sure.
So are you saying that our grade isn't accurate?
The individual grades for an evaluation should be accurate. So if you got a 75% or whatever it is, that should be accurate. But the way that the Brightspace calculates your final grade, it's not accurate. And you don't want it to calculate it that way. So is that clear?
Yeah.
Right, so over the next couple of days, I'm going to be doing that work, and if you want, I can talk to you individually about your grade. Right? I won't talk to you in groups about your grades. So in terms of now this week, we have lecture today, we have lecture tomorrow, and we'll see how it goes on Wednesday. But we have 2 more topics here. The SNS is the somatic nervous system. And the somatic nervous system consists of 2 parts there. There's an efferent system and an afferent system, and we've already talked a lot about the efferent system. The efferent system consists of 2 main components, and that is to say there's neuromuscular junctions and then there's neuroglandular junctions. Those neuroglandular junctions are not something that we'll talk about for this semester. We'll just sort of assume that they're like neuromuscular junctions. The other part of it is the afferent system, and the afferent system is what we're going to be talking about today. It's weird because these 2 chapters that we're going to do in 2 days or 2 or 3 days, these 2 chapters are pretty short, but they're incredibly complex. They affect every part of your waking life and your non-waking life, and they're really, like I said, they're really complex. And in terms of the anatomy and the physiology, there's not a lot new there. We'll talk about some of that new material today. There's not a lot of new there, but it's like I said, it's just it affects you everyday life. one of the things when I talk about afferent nervous system of the SNS, the sensory nervous system, the somatic nervous system, I'm talking about sensation and how you perceive things like pain. If any of you know anyone that has chronic pain, I know a few years ago I had a problem. I had basically a chronic headache, and found out that I had a infected tooth, and then I got a root canal, and then it fixed it. Like, it was incredible how good everything felt after that. But it was just, I was in a crappy mood. I wasn't happy at work. I wasn't happy at home. I was short with everybody, and just sort of a mean old grumpy guy, more than I am normally. And that was caused from chronic pain, and that was a simple acute thing to fix. Chronic pain can be just debilitating, and it's part of the somatic nervous system. So let's talk a little bit about the somatic nervous system. The other thing to remember is, now in addition to when we talk about the somatic nervous system, remember when we talk about the action potentials, all of the terminology associated with Unit 4 material, associated with neurons, neuroglia. In far as the somatic nervous system, we're talking about the peripheral nervous system. And if we're talking about the peripheral nervous system, then we're talking about special types of neuroglia as opposed to CNS or central nervous system neuroglia. When you're talking about sensation here, the sensation is the process sort of initiated by the sensory receptors, and they send, as I said already, they send afferent signals to the CNS, and those afferent signals come from the periphery, regardless of where they are in the body. If they're in the abdominal pelvic region, it's still part of the somatic nervous system. They can be directly adjacent to the spinal cord, and they're still part of the somatic nervous system. Perception is the brain's sort of recognition that pain is occurring, or that there's sensation occurring. So that the brain is receiving information before you are aware of it. It's receiving information, and so that perception is something that is different than we're talking about today. The information is being sent via the afferent pathway to the brain. Whether the brain recognizes it or not, or chooses to react to it or not, is a different issue. And then finally here, I'm going to talk about different types of senses. We're talking about general senses in this class, and then in AMP2, they're going to talk about special senses. And special senses is a whole other ball of wax, whole other type of information. In terms of general senses here, we have somatic and visceral senses, and those somatic and visceral senses tend to be either visceral being internal organs, and somatic including generally the skin, the muscles, and it sort of the visceral gets into the musculature too. But somatic information, we'll talk about pressure, sensory structures associated with touch, pressure, temperature, proprioception, and pain. And those individual receptors here respond to individual stimuli at a particular way. And again, they synapse with these are modified epithelial tissue cells, and these modified epithelial cells send action potentials, cause these dendrites to exceed the threshold, and then the neurons send signals. When you see the word special senses here, first of all, the special senses here, including smell and taste, vision and hearing, those are the main ones. And it says also that there's balance, but we're going to be talking about balance in the context of this class. Balance is part of the general senses here in my book, also in this book, and I'm not sure why they're talking about it as a special sense. When you start organizing all of this here, we're talking about organizing all different types of senses, either into general or special senses here. And on the right-hand side here, we have these first four are not relevant to this class. And you're going to spend an awful lot of time in AMP2, things on structures of the vestibulocochlear structure, the eye, the retina, the layers of the retina, and so on. Balance we'll talk about today, or this week. So what we'll really focus on though is somatic sensory and visceral sensory, and those just basically are talking about the source of information. Where is that information coming from? Regardless of where it's coming from, it's always going to the same place, and that is to say it's going to the central nervous system via the spinal cord and then the brain. Now, when we start talking about individual sensory receptors here, we have a number of different types here. And on the left-hand side, we have some general sensory structures here, visceral, including visceral and somatic. And then at the bottom here are special senses. Those special senses, again, we're not worried about. If you look at the receptor type, this is really what I want you to focus on here. So in terms of the receptor type, there's mechanoreceptors, there's thermoreceptors, and then there's nociceptors or pain receptors. So those nociceptors or pain receptors are, those are important structures. And those, depending on where the structure is, it can either be somatic sensory or visceral sensory. So those are the three of the main structures that we'll talk about from a receptor type. We're also going to talk about chemoreceptors that are not in context with the special senses. So a chemoreceptor is simply detecting specialized chemicals in the body. And in this case, the chemoreceptors in special senses is talking about the special sense of smell. So I'm not talking about chemoreception in that context. I'm talking about chemoreception in a very different context. And then on the finally, on the right-hand side here, some specific examples here. If you look at some of these specific examples here, especially the first group right here, these first group are the ones that I want you to, that I'm going to spend time on. So the Merkel disc, Pacinian corpuscles, Meissner corpuscles, free nerve endings, and so on. Those things are what we're going to be focused on today. So in terms of sensory receptors here, so I mentioned sensory receptors, I mentioned chemoreceptors. Chemoreceptors is these sensory structures that respond to individual specialized chemicals, and those specialized chemicals then will initiate an action potential if it exceeds a threshold. When that is associated with smell, then that's not subject for our topic here. But when it's found within the body, it is something that we'll talk about. In addition to chemoreceptors, there's mechanoreceptors, and they come in a couple different varieties. Here, there's touch, pressure, and proprioception. Proprioception is balance and maintenance of the internal joints in our appendages. So proprioception is associated with individual joints and articulations. Pressure sensors, mechanoreceptors come in a couple of different varieties, and those will become more evident in a little bit. One in particular is called a baroreceptor or a stretch receptor. That is, there's a couple of different ways. If you push one of your fingers onto your arm, that you can feel your arm getting pushed on, and that's one form of pressure. But you can also have internal pressure, and that internal pressure would be causing stretch in your stomach, for instance. So you guys just ate breakfast, your stomach is now getting pushed out, and if you keep eating and keep eating and keep eating, right, that stomach is going to get pushed out more and more and more, and that's stretch receptors. Your stomach has stretch receptors, and that's going to tell the body to start doing something else. And then again, these nociceptors are another important part of our body. Depending on where they're located here, we can talk about these receptors either being visceral or cutaneous. So cutaneous tends to be superficial. Proprioceptors have joints and tendons, and there's stretch receptors there. Again, that's what we're talking about here. We're not talking about balance from the context of the vestibulocochlear nerve, that cranial nerve that we talked about last week, or for this quiz. But proprioceptors are associated with joints and articulations. In terms of the types of sensory receptors here. Here's a list, and I'm going to get into them in a minute. But all of these structures are important, and we'll have questions on in the exam about their structure, what they look like, where they're found, and what their function is. Oops. So those classification of general sensory structures are here in this slide right here. If you look at the on the left-hand side, these are, so there's, let me see something here. I forget what so there's 2, 4, 6, 8 total. In this first slide here, I've just talked about 6 of them. What I did here is that there's some there's terms that are synonymous with one another. So you may see me or hear me talking about Merkel disc or tactile discs, hair follicle receptors or root hair plexus, Pacinian corpuscles or lamellar corpuscles, Ruffinian end organs or bulbous corpuscles, and Meissner corpuscles or tactile corpuscles. Those are synonymous with one another. Starting with the last one first here, those Meissner corpuscles, so those tactile corpuscles are really interesting for me. There's a whole lab that I had designed that we just don't have time to do this semester, but they're basically trying to illustrate, trying to get you to understand how depending on where you're getting touched on your body, the Meissner corpuscles are either infrequent or absent, or very, very frequent and very abundant. So those Meissner corpuscles will do something called 2-point discrimination. And 2-point discrimination is very simply illustrated here. If you have 2 things that are touching your hand and you get closer and closer, you can tell, even with your eyes closed, that I have 2 things touching my middle finger here, because again, this is my favorite appendage. I can tell that my fingers are touching my middle finger there, and I can say that there's, even if it's a couple of millimeters away, I can tell the difference here. So our hands have a lot of these Meissner or tactile corpuscles. In contrast, if I do that on my back, and you probably all had this occur, you go up to someone, to your mom or someone, your dad, some other family member, I got an itch on my back. Can you scratch my back? Not right there. No, not right there. No, over, up, down. Oh, okay, right there. And you have no idea where it is generally, because you don't have a lot of these Meissner corpuscles on your back. So that is an illustration of how that works here, the sensory structures here. So on our back, we need to know that there's something on there. We need to know that there's a bug there, or whatever it is. Right? But we don't need to know so much detail as we do on our hands, because we're so able to use our hands so much. If you just go up from there, there's these individual sensory structures are located in different layers of the skin. And so, for instance, these Ruffinian end organs are deep, and the Pacinian corpuscles are as well deep. Both those respond to pressure or touch here. There are hair follicle receptors here, and I still have them in case you were wondering. Their hair follicle receptors here are associated with superficial portion of the skin. Again, those will be for that, and along with Merkel disc will be light touch. And then finally here, there's free nerve endings that have a number of different functions there, but generally it's pain and itch that are most common sort of descriptors. The last 2 that on this list here are the muscle spindles and the Golgi tendon organs. The muscle spindles there are associated with stretch receptors, and have I talked about stretch receptors already? Yes, I have. That's in the context of reflexes and monosynaptic reflexes. So when you're talking about muscle spindles and monosynaptic reflex arcs, right? So there's a question, there's a way that I can ask this material from Unit 4 here So there's overlap again. And then similarly, these Golgi tendon organs there are associated with the stretch of the tendon and associated with the muscle contraction. Again, that would also be something with the reflexes. Now, when I talk about proprioception here, though, once again, I'm talking about am I standing on one leg, not am I about to fall over. If I'm standing on one leg and I'm about to fall over, that's a whole other part of our body that is trying to figure out what the hell's happening. That's part of our vestibulocochlear nerve, and that's balance and hearing. But what I'm talking about when I'm talking about proprioception is what's happening at my right knee, what's happening on my right hip as I pull up my leg there, and I'm only worried about what's happening there. So I'm artificially separating some of this stuff. Here is a picture of this where the location is, and if you just to orient you from this, so here is the epidermis, the dermis, there's the main part is the reticular dermis here, and then this is the papillary dermis right here. So these are the dermal papillae, right? And so you should remember, if I were to use the term dermal papillae that are two-point Meissner corpuscles are involved in two-point discrimination. They're located in dermal papillae, so they're part of the dermis and not part of the epidermis. That is an important recognition, right? So bless you. If you don't remember what dermal papillae are, right? So reacquaint yourself with that. In terms of hair follicles, receptors there, they're associated with the base of the hair follicle, and that base of the hair follicle again is a part of the epidermis because the epidermis has penetrated deep into the dermis here, down almost to the hypodermis, but it is still part of the epidermis. There's Merkel discs and free nerve endings that are also superficial. They're not necessarily associated with the dermal papillae necessarily, but they're at that level. They're superficial and they're responding to pressure and pain. And then the two that are deepest are the Pacinian corpuscle and the Ruffinian end organ. And again, those are going to be responsible for pressure within within joints, and they are deep. So depending on where you are in your body, you're going to have a greater concentration of one set of sensory receptors or another set of sensory receptors. Again, depending on where you are. So now I'm just going to go through these 8 different these nerve endings here, or these sensory structures, and we'll talk about generally what their function is. These free nerve endings are the first of this group. This is the most common one, and pain, temperature, and itch. I'm not going to ask about thermoreceptors because I'm trying to for free nerve endings because I'm trying to separate thermoreception with other structures here. But in particular here, pain and itch are 2 examples of things that are free nerve endings sense. Those epithelial tissues, modified epithelial tissues, are scattered throughout. Those structures there are scattered throughout the epithelial and then into the connective tissue. And they will respond to itch and to temperature. A few years ago, I had a pretty bad case of poison ivy, and so I put sort of lotion on it to try to calm the itch, and so my free nerve endings were there. But then I hit on the idea of taking a hot shower, a really, really hot shower. And I had it on my right arm, and I would just basically pour the hot water, let the hot water hit all that area where the poison ivy was, and so those itch receptors then sensed heat and pain, and then the itch receptors then were overwhelmed by those pain receptors, and then it stopped. It sort of temporarily stopped it. My wife thought I was crazy, but it worked for me. But she also thinks I'm crazy about general stuff too. An example of light touch is Merkel or tactile discs, rather. Again, these are superficial in nature. They're for the basal layers of the epidermis. Again, they're modified epithelial cells. Obvious one, hair follicle receptors there. they're sensitive to when they're moved, right, to any type of pressure associated with that. So moving the hair, you can definitely sense it, right? The end organ here, what this means here, the receptors overlap, those receptor fields overlap. That means that because they're overlapping, you can tell that the hair or the hairs are moving, but you can't tell which hairs are moving. In other words, you could say that it's on the right side of your head, but you can't say, oh, it's this one right here, or it's that one right there. It's a little bit like being able to distinguish two points touching your hand. You can definitely distinguish that. When receptor fields overlap, it's hard for you to distinguish the source of the information or the structure. These two are in the tend to be deeper in the body. They're Pacinian corpuscles, and I will talk about the lamellar corpuscles. That's the other synonym. I won't use lamellated. These are deep in the dermis or the hypodermis. I will say they're in the dermis, just to make it clear. And again, there's deep cutaneous pressure or vibration. That's the main thing. And they're associated with joints here. Ruffinian end organs are responsible for touch or pressure, tend to be in the fingers. And so here. And if you look at this picture here, this sort of illustrates a part of the lab that I was going to have you guys do. Two-point discrimination is the ability to determine pressure or some other type of sensory issue simultaneously and depending on how far they are apart. On the back, this is the person on the right-hand side. They can't detect, it might be 64 millimeters or 6.4 centimeters away, and they still can't detect that they're different, two different things hitting the body. But in the hand and then on the tongue, you can touch, you can detect those things. So the ability to detect simultaneous structures in the skin is a really important adaptation for us. So if you're eating something and you know as well as I do, right? You're eating something and there's something that's funny in your mouth and you're like, oh, what the heck is that? And you sort of roll it around a little bit and then you take it out. So my daughter was talking about, so I don't know who was talking about, someone was talking about roommates who were interesting. And I had a graduate student, friend of mine who had just come from China, and he was sitting there eating, I went to pick him up, and he was sitting there eating breakfast, and he had something in his mouth that he didn't want, and so he went, and he just spit it on the floor. I was like, Shashu, what are you doing? He's like, oh, I'm sorry. And so anyway, I was like, oh, you probably shouldn't do that. I was trying to give him an idea about our customs, because he was literally here for like a month, and he barely spoke English. But one of the things that he did was he didn't think it was a big deal. He was in his house to spit on the floor. I was like, you probably shouldn't do that if you're not in your house. I didn't want him to get embarrassed or anything like that. But that ability to identify something that is either something that you want to swallow, something you don't want to swallow, is partly a result of these Meissner corpuscles, these tactile corpuscles. And then muscle spindles are an example of things that are associated with individual joints and articulations. They provide information about the length of the muscle, and what's the length of the muscle got to do. It's whether or not it's contracted or partially contracted or completely relaxed. And postural muscles, stretch reflex. Again, these things right here, stretch reflex. What type of reflex are we talking about here? We're talking about either polysynaptic or monosynaptic reflex arcs. And if you look at this diagram before I go here, remember here, reflexes involve especially monosynaptic reflex arcs. reflexes involve both sensory and motor neurons that are sending signals from an area to an area, and in the case of the monosynaptic reflex, it's sending a signal from that area and then to that area, essentially the same area. And then Golgi tendon organs will help protect the structure of the muscle so that it doesn't allow for the tension on the tendon to be too much, to get too much. And if there is too much, then the body's going to try to do as best it can to protect that muscle or that joint. So the Golgi tendon organs will help with that. So that sort of gives you an overview of some of the most abundant, most common sensory structures, sensory receptors there. And now we're going to get into graded or different types of potentials here. And when I talk about potentials here, we're talking about action potentials in a neuron. We're talking about all the different terminology associated with neurons and action potentials, be it the axon lemma or the axon hillock or anything like that. When you're talking about receptor potential here, the resting membrane potential there is at negative 70 millivolts here, and that graded potential will increase a little bit and increase a little bit and increase a little bit until it exceeds the threshold, and then it sends that action potential there. There are different types of receptors that we have in our body. There's primary and secondary receptors there. Those primary receptors are going to be the ones that are responding to individual stimuli, and secondary receptors are the ones that send signals further down the line. Those secondary receptors cause the release of neurotransmitters. Now, don't worry about the fact that I can ask about the different types of receptors, but I won't ask for examples of secondary receptors, especially in the context of smell, taste, hearing, and balance, because those are special senses. So once again, I'm trying to sort of draw that line right there. So one example of a sensory receptor here is a mechanoreceptor called a Pacinian corpuscle. So now I can ask you which of the following are mechanoreceptors or which of the following are not mechanoreceptors, whatever the case might be. And one example of that is a Pacinian corpuscle. It is going to be responding to individuals' pressure right there, and then it sends signals down the dendrite, down the action potential rather, down the neuron to the terminal axon, right? And that's sending it towards the central nervous system in an afferent direction. If you look at the structure of this neuron right here, by the way, look at this here. So this is part of the peripheral nervous system. So this is a unipolar or a pseudo-unipolar neuron, and that's another question that I can ask and can filter in there. And how do I know that? Because the soma or the cell body is away from the dendrite and the axon axis there, the main part of that neuron. And so the action potential is going from left to right, and it is nearly completely skipping the soma or the cell body. In terms of the distinction here between primary and secondary receptors here is an example of a secondary receptor. Basically, this modified epithelial cell sends an exon that is associated with taste in this case, and it sends a signal via a neurotransmitter to a neuron. I'll ask you about the general structure here, but I won't ask you for specific examples. So I just mentioned chemoreceptors, and I've mentioned chemoreceptors several times over the course of this portion of the lecture. And I said that chemoreceptors are associated with special senses, and we won't talk about them unless we talk about chemoreceptors here. So chemoreceptors are not chemoreceptors. So if your younger sibling produces a noxious odor in your house, that is chemoreception, and that is not what we're talking about here. What we're talking about here is chemoreception in the context of two very, very important chemicals, and it's got nothing to do with your nose. It's got internal organs here. So these are visceral receptors, here, and visceral receptors here are not associated with the superficial portion of the body. Right? So there's two examples there. There's carotid bodies and aortic bodies here. A carotid body, we have a carotid artery, paired carotid arteries that go up on either side of our neck that come up from the heart and that are feeding blood to the brain. That's one example. And then there's aortic bodies, and an aortic body is an area of the arch of the aorta, which I don't really care if you know that, but they're right off of the left ventricle. And basically what they sense is, what is the carbon dioxide content of the blood? If there's high carbon dioxide content in the blood going to the brain, that's a problem. If there's high carbon dioxide content leaving the heart, that's a problem. What's the solution there? Well, there's a couple of different mechanisms there, but that's the first detection there. Another thing that they will do is these chemoreceptors will also respond to amounts of water in your body. And so if there's lack of water in your body, then what your body does is it doesn't control water very well, but I don't know if I've ever mentioned the sodium potassium pump. Our bodies are quite able to control sodium levels. And so what we do is what our body does in areas of like the urinary system. If we need water in our body, maintained in our body, and so we want to reduce the amount of urine in our urine output, what we do is we suck up some sodium here, and water follows the sodium. And so what's that process? That's just process of diffusion. So there's a couple of different ways of examples of chemoreceptions. And so in terms of the first two, carotid bodies and aortic bodies here, what are they measuring here? They really want to measure oxygen, because oxygen is the thing that's really important here. But what we measure here is carbon dioxide and pH. When you have lots of carbon dioxide dissolved in water, it produces a weak acid. And so as a result of that, the lower the acidic content of your blood, and the higher the concentration of carbon dioxide in the blood, means that there's a lower concentration of oxygen in the blood. So it is more, our bodies are more interested in this really critical chemical than oxygen, but it uses these two as a proxy, pH and carbon dioxide. So these carotid and aortic bodies there are associated with individual cranial nerves. And if you look on the right-hand side, the carotid bodies are associated with the cranial nerve 9, and the aortic bodies, cranial nerve 10. Those cranial nerves, they're associated with either the respiratory or cardiovascular centers of the brain. And so as a result, where are they going to be going to? They're going to be going to the medulla oblongata. So in other words, they're going to be sending signals from these two areas right here, sending it to the medulla. And so these brain and respiratory centers are up right here. The respiratory center is right here in the medulla. So these centers here will, when pH decreases and when carbon dioxide increases, those two things happen simultaneously, because pH, as it goes down, acidic content increases. When those things happen, then respiratory rate will increase, and also heart rate will increase. Another sensory receptor that is deep, a different type, is a baroreceptor. And a baroreceptor is a pressure receptor. And I mentioned pressure receptors in the context of the digestive system, for instance, in the stomach. Right? So a baroreceptor is a pressure receptor, and basically if you imagine you're blowing up a balloon, and it has a little bit of pressure on it, and you blow it more and more and more until it pops, right? So that's too much right here. There's 6 examples of baroreceptors in our body. Some of them are associated with things that we're not going to worry about too much here. Again, now the carotid and aortic sinuses, that's entirely everything you need to know about the circulatory system for this class. Again, they're part of associated with the neck and right off of the left ventricle of the heart. Those sinuses will detect pressure, blood pressure in those areas here. So not only are they measuring chemical receptors or chemoreceptors, there's also baroreceptors. If the pressure is low in going to the brain and the carbon dioxide content is high, that's a bad combination. So as a result, the body has to do something different. It's going to send signals via the cranial nerves to the medulla oblongata, and the respiratory center is going to increase, the cardiovascular center is going to increase, and that pumping rate is going to increase. So not only do we have chemoreceptors right there, we also have these pressure receptors called baroreceptors. When those baroreceptors, when the signal from the brain says to increase respiratory rate, you don't want to increase it too much, but you want to increase it enough so that there's enough oxygen, or there's an appropriate amount of oxygen going to the body, to the brain especially. And so there's pressure receptors or baroreceptors in the lungs. And so I definitely want you to recognize individual receptors as a chronic sinus, an aortic sinus, or the lungs. So those three are specific. The digestive system or the digestive tract and the colon are the same thing for this class. They're different. Right? I'm just trying to simplify it. Generally speaking, the digestive tract is early on and the colon is later on in the digestive system here. And then the urinary system is a different thing all completely, and it is associated with cleaning the blood. So the bladder or the urinary system here is associated with the kidneys. And again, that's what I was talking about when we're talking about movement of water, either retaining water or not retaining water in our bodies. So there's 6 baroreceptors in the body generally here, but I will only ask about 5. So I won't ask you to distinguish between the digestive tract and the colon. So you can just talk about those two as the same thing. Depending on where we are in the body, these receptors will respond to things differently. And as they respond to things differently, things can change over time. So I was telling you a minute ago about me getting poison ivy and putting really, really hot water on my skin. Those sensory structures, when it was really itchy, they were sending signals there and causing me to really, really want to itch my arm. And when I put hot water on it, those itch receptors that were then overwhelmed and things changed, that's called adaptation. That decreased sensitivity to a stimulus continued, and that adaptation of those neurons or those sensory structures then changed the signal from itch to pain to relief for me. And that's called adaptation. We talk about proprioceptors, and those are part of, and so adaptation is when the sensory structures change over time to the same stimulus. Proprioceptors, there are one example of movement of the body. You can close your eyes and you can kick out your right foot, and you know where your right foot is without actually looking at it. And so that's the ability of our body is called proprioception. There are different types of receptors there that either respond to stimuli here. There is either tonic response or a phasic response. Tonic response will adapt or accommodate very slowly, and phasic response are able to do it very rapidly. So they respond to these stimuli differently. And so first of all, we're going to talk about this picture right here. This is an example of a tonic receptor or a tonic response. And if you look at the bottom, this red area right here, these are a series of action potentials here. So once again, now this is set the resting membrane potential. If I were to mention that, the resting membrane potential where my red dot is negative 70 millivolts. That exceeds the threshold. It causes the action potential. There is hyperstimulation right there, hyperstimulation, and then it returns to resting membrane potential. So now this at the top here is when you have the sensory structures that at a certain level here that is non-stimulated, and so these things will send signals periodically. There's no information coming, no information coming, no information coming, until there's information coming, and then they respond very, very rapidly. These tonic receptors are always active, and they're always sending signals until there's a response, and then they send signals a lot more. one example of that is the pain receptors. So you have those pain receptors, whether you have pain or not, are sending signals to your body, and they're sending, basically they're saying there's no pain from this area of the body, there's no pain from that area of the body. So tonic receptors are always active. Phasic receptors respond very, very rapidly here, and these are normally inactive. And so, in other words, these things, this is different here. So, two examples there include Pacini and Meissner corpuscles there, and here is again the frequency of the action potentials, and you can see the difference here in during when it's not being stimulated here. There's no activity. Then there's a little bit of sensitivity. The stimulus increases to a certain level. There's activity when it either changes here. So we're talking about phasic receptors. Look at change in the sensitivity. When there's increase, there's then action potentials sending signals here, and then there's a change. Then there's a stabilization of the pressure or the stimulus, and that pressure, the action potentials go down. And then when the sensory structure then decreases, or the pressure, the stimulus decreases here, those changes then respond again. So in other words, the phasic receptors here respond to a change in pressure, either a big increase or a decrease in pressure. And these things respond strongly at first, but then decrease over time, and that's indeed what's being shown here. So there's no response, then there's a response, and then there's no response, and then there's a response. And there's only a response when it's either increasing or decreasing. So those are talking about the overall general structure of the sensory structures in the body. This portion of the lecture is associated with the combination of afferent nervous system here, so sensory structures. Where do those sensory structures send signals to? They're sending it generally to the brain. How do they get there? Through the spinal cord. Where on the spinal cord do they go? And so when we talk about the spinal cord here, here's a cross section of the spinal cord with the anterior median fissure. I can ask about that. I could ask about the dorsal root ganglion. We can talk about the ventral root or the motor, the ventral root or the dorsal root. One is motor, one is sensory. The dorsal root is sensory right here. And when you're sending these signals there from the periphery to the brain, these afferent signals there have to, and we've already talked about this, right? We've talked about ascending pathways or ascending tracks, and we're not talking about descending tracks here. So we're only interested in ascending tracks because that's the focus of this topic here. These are a series of these green areas right here in the white matter of the spinal cord. These green areas right here are individual tracts, and those individual tracts have specialized names in it. And if you look at the bottom here, the first half of the word indicates the origin, and the second half indicates the termination. What does termination mean? It means where does it end? Where does the signal end? And we're talking about in the brain. So where does it originate here? So there's the anterior, the posterior spinocerebellar tract. So the spinocerebellar tract here, we're talking about somewhere associated with the spinal column here and the cerebellum. The spinothalamic tract, where is it going to terminate? In the thalamus. What's the thalamus going to do? The thalamus is going to determine, basically it's a clearinghouse for information, and that the thalamus is associated with basically figuring out what to do to these stimuli here. So these individual ascending pathways or these ascending tracts are entirely white matter here, and for the purposes of this discussion, we're not worried about descending tracts or motor pathways. So the first of these is that there's the spinothalamic tract. So where does it originate? Well, it originates in any of the structures that we've already talked about. Any of those general sensory structures we already talked about, whether it's pain or pressure, temperature, or baroreception otherwise, and things like that. So that from there, it's going to go to the posterior horn of the spinal cord. How do I know that? Because that's the dorsal root is the sensory structure. So it's going to go to the dorsal horn or the posterior horn. And from the posterior horn is gray matter. Then there's going to be a synapse with interneurons, these specialized interneurons, that will send signals in multiple directions. One direction will send it from one side of the gray matter to the other side, and then they will send signals to the spinal thalamic tract. That spinal thalamic tract, that white matter then, will be the ascension, or the will ascend or move superiorly to the thalamus, and then there's individual neurons within the thalamus then that will that are associated with sensation. And then once again, the thalamus, we've talked about the brain already. The thalamus is an incredible source of sort of figuring out whether or not to respond to specialized stimuli, and the somatic sensory cortex, there's specialized areas of the thalamus that just deal with this information. So the spinal thalamic tract is one example of that, and so that it's going to terminate in the thalamus. And where does it originate? Depending on wherever the sensory structure is being stimulated. So it could be the hands, it could be the feet, it could be the back, it could be the face. Okay, so anywhere in the body, you can send signals via the spinal thalamic tract. There's another example here, the trigeminal thalamic tract. And so what is the trigeminal thalamic tract here? Well, it's cranial nerve number 5. So now here's a way that I can link the cranial nerves and this discussion here. Now, the cranial nerve, the trigeminal nerve is a mixed nerve, so there's motor aspects of the trigeminal nerve as well, but I'm not worried about those. For this one, we're just worried about sensory structures here. These cranial nerves there, the secondary neurons there run from the, they run to the brain via the trigeminal nerve. And they carry similar information to the spinal thalamic information, but the trigeminal tract, trigeminal thalamic tract, is going to terminate in the same place as the spinal thalamic in the thalamus, but it's going to originate generally in the face. Spinocerebellar tract, again, where are we starting? Again, wherever in the body that the sensory structure is stimulated here. And now, how is the cerebellar information different than the thalamic information? The thalamic information is particularly information associated with sensory structures that are not related to movement. The cerebellum is associated with movement, is focused on movement and balance. And so proprioception then is where the spinal cerebellar tract will send signals. So the spinal cerebellar tracts there will send signals to the cerebellum, and there are proprioceptions there. Again, so for instance, there's the Ruffini end organ that is associated with the stretch receptors within a muscle, and they will send signals to the spinal cord, and then the cerebellum telling your body, oh, you're standing on your right leg, you have your left leg up. Right? So obviously that's unconscious. Right here, you don't think about that, you just do it. And the other part about this is when you're talking about actual movements here, actual movements are controlled also by the vestibulocochlear nerve, and there's dynamic and static equilibrium. So we talked about dynamic and static equilibrium generally. I won't get into any great detail about them. What is the difference between those two? When we talked about the cranial nerve number 8, dynamic equilibrium is the equilibrium of balance when you're moving, and static equilibrium is the equilibrium of your body when you're not moving, when you're sitting down, for instance. And again, I talked about the structure when you're moving and you sense movement and you don't feel like you should be moving. What is a common response to that? It can either be seasickness or car sickness. So that is when the spinal cerebellar tract there is a different set of structure that's associated with the cranial nerves. So again, now this is absent of the cranial nerves here, and it's just through the body to the cerebellum. Pain pathways is a generalized area. Pain, sensory structures there are most abundant there in the body. I've talked about chronic pain. Chronic pain in general is an unpleasant signal that triggers a number of different responses. It can be long-term responses, autonomic responses, and it can produce psychological responses as well. So you could get, if you think that someone is going to hurt you, you can respond to that and say, before the stimulus is applied, and you can respond to that even before it happens. So that there's different types of signals there. There could be sharp localized pain that are acute, and then there's also more slowly propagated action potentials that are just slower that are not quite not acute, but they're more associated with chronic associations or chronic pain. Burning, aching pain is an example of chronic pain, and that's an example of when someone gets old. I won't tell you who I'm thinking about. But pain relieving medications there have a number of different ways to respond to stimuli, or to help not respond to stimuli, but these analgesics here help the body respond to pain. And one of the things that they do is they send signals to the thalamus here, and they suppress action potentials within the thalamus. So the sensory structure is still sending signals, but the thalamus is not responding to those signals. The thalamus is not saying, your body is in pain at this joint or this point in your body. This pain relieving structure is called analgesia. Another example of pain is referred pain, and referred pain is simply when you have one region of the body that's not the source of the pain, or the source of the stimulus, but you have pain there, and there's a great deal of complexity associated with referred pain. Sometimes you have referred pain when you have an amputation and someone has a limb that's amputated, and they say, I have pain in that limb and I know that limb is missing. That's all of that referred pain is associated with the brain, and particularly with the thalamus. I talked a little bit about chronic and acute pain here. Acute pain is when you have a significant injury that's obvious. So we've broken a bone, there's an acute example there, and chronic is, it's just a general term right here. So for instance, you could have tissue damage from arthritis. Now, why would I bring up arthritis here? Well, have I ever talked about arthritis? Yes, I have. We talked about the structure, the degradation of individual joints and articulations here. That can lead to chronic pain. There's other things that I would be less interested in talking about. So migraines, depression, frustration, hopelessness, all can lead to chronic pain as well. But those are not nearly as simple, and I like to focus on simple things. So for instance, chronic pain could be associated with arthritis. And so what I want to do now is I want to end this lecture here. We've just talked about the somatic nervous system, all the different sensory structures associated with it. And for the final topic here, we'll talk about the autonomic nervous system, and we'll see how where we go tomorrow. Okay? So if you guys have any questions about your grades or anything like that, or about the quiz today, you can feel free to stop by. I want to make sure that I'll just talk to you individually. But otherwise, I'm done talking today, and I will see you tomorrow.